Good evening, everyone. This side Dr. Abhijeet, and today we are going to discuss another topic that is the carbohydrate chemistry part 2. The Oracle has initiated a 21 days live program, right, where we are going to cover the entire no syllabus for the first prof exam. And I will be revising the most important questions that are no relevant with the topic and that are most important that can be asked in your upcoming exam, right? So that today the questions that I will be covering, yeah, the 10 marks question, the 10 marks question. Yes, the biochemical significance of glycoproteins and proteoglycans in cellular functions. The short essays, which are like 8 to 5 marks question, either 8 marks or 5 marks. I will be discussing the glycolipids, the role in the cell membrane and cell signaling, the biological importance of glycosaminoglycans. Very, very important question. I'll ask every student to put a dollar sign over here. As you all know, dollars are very, very important. So yes, I'm going to discuss the concept of lectins and what's their role and function in the cell-to-cell interaction. I will be also covering the short answer questions. I mean, 3 marks or 2 marks question. I will be discussing the blood group antigens and how they are related to carbohydrates, right? I will be discussing the term glycocalyx and what its role in the extracellular matrix. And I will be also focusing on how the carbohydrates are involved in cell addition, right? So yes. Now, before starting with this, guys, I just wanted you to, yeah, as we all know about the Oracle app, right? Yes. So as we all know about the Oracle app, guys, we have the recorded classes for flexible learning. If you guys are not attending the classes live, you can always go on for the recorded version that is available on the app. The live classes are conducted daily between 6 p.m. to 10 p.m. and you all guys can benefit from these live classes, right? Apart from that, if you want to go through the practical session videos, they are also like available on the app. The recorded version is there. Apart from this, under the express exam module, under 20 hours, we have covered the entire like the most important questions that can be asked in biochemistry, the most important topics, the most repeated previous year topics are being covered over here, and you guys can go through these exam modules also. And you can also go through the hard copy book set, right? Which is now available at your doorstep. You can order it. And these hard copy book set basically consist of concise notes, in the form of question and answer. The exact way that you have to write the answer in your prof exams, right? They also have fun riddles, quiz, MCQs, right? And problem solving assessment for you people. So we have the foundational classes for flexible learning, right? We have the live classes. We have the express exam module, which covers everything under 20 hours, right? We have one-to-one doubt clearing session. If you guys have any doubt, you can just go on for clearing your doubt by booking a prior appointment with the faculty, right? And you can get all your doubts cleared. Hard copy book set is also available, right? You already know they are actually curated by the experienced faculties. No, they have included the points which are to be, which are important and that has to be mentioned in the right exam paper, right? And yes, there are MCQs, there are fun quizzes, riddles, everything they are like included in the book itself. Question bank basically consists of questions from basic to advanced level. After each and every topic, you will get questions from basic to advanced level. They also cover questions from the national exams like the NEET PG, the INI CET exam, also the FMG exam. The past 10 to 15-year questions are being covered. Even basically the hot favorite of examiner nowadays, that is the clinical vignettes, they are also being covered over here. The practical sessions, basically we have these practical recorded videos which are available on the app. You can just go on for the recorded version. And we have the subscription plans, the foundation plan, we have the advanced learning plan, we have the complete learning plan, and we have the ultimate MBBS plan. You can use my code Dr. Abhijeet 20 for additional discounts. Right? So yes, let's start with the today's topic. Hmm. Now, the very first question, they are asking about, yes, the biochemical significance of glycoproteins and proteoglycans. Now, first, let us just talk about the proteoglycans. Now, when I say proteoglycans, guys, when I say proteoglycans, so let me just write it over here. Proteoglycans. Now, proteoglycans is basically formed from proteins, and we have glycans, that is the glycosaminoglycans. This glycosaminoglycans, it is a heteropolysaccharide that we discussed yesterday, right? So this GAG basically stands for glycosaminoglycans, that is GAG. Okay, it is a type of heteropolysaccharide, guys. You already know it's a type of heteropolysaccharide. I will be discussing this question later. Yes, it's a type of heteropolysaccharide, right? Now, glycosaminoglycans is basically, yeah, it is basically composed of no this carbohydrate group. We have the carbohydrate group. We have amino sugar. Amino glycans means the sugar. Yes, the amino sugar. So remember, guys, the glycosaminoglycans. Okay, the glycosaminoglycans. It is basically a long. Remember, guys, it is a long, unbranched, unbranched heteropolysaccharide. Heteropolysaccharide. And remember, it has a repeating disaccharide unit. It has a repeating disaccharide unit. Now, when I say disaccharide, guys, we have already discussed the disaccharides yesterday, right? So we have maltose, lactose, sucrose. No, these are the various disaccharides. Now, this disaccharide it is basically composed of two components. It is composed of two components, guys. Remember, we have the amino sugar. We have the amino sugar, and we have the acidic sugar. We have the amino sugar and we have the acidic sugar, right? Amino sugar is basically when an amine group it is joined with the sugar. So for example, guys, you are much aware of glucosamine. Now, when I say glucosamine, it is glucose plus amine sugar, NH2 group attached to it. Or I can have another one that is galactosamine. Galactosamine. It is galactose plus amine sugar. When I say acidic sugar, guys, I am actually talking about, yes, glucuronic acid, glucuronic acid, or either it can be iduronic acid. Once we have discussed the iduronic acid, we have discussed the oxidation of no sugars. The aldehyde group it is oxidized to the acid. Okay, the COOH group. That's why glucose when oxidized, right? It forms glucuronic acid. The first carbon that is carrying the functional group that is CHO, it will be changed into COOH. Right, so that is the glycosaminoglycans. So proteoglycan means protein plus glycosaminoglycans. Proteins plus glycosaminoglycans. Now, when I say in proteoglycans, what will be the protein percentage? Remember, it is 5%, and glycosaminoglycan it is. I mean, sorry, protein is 95. Sorry. Yeah, okay. Let me write like this. Yeah, protein is 95% and this glycosaminoglycans, okay, it is basically, no, it was right. Just a moment. The protein is 5% and yes. The glycosaminoglycans, it is 95%. 95%. Now, exactly opposite, guys, if we talk about glycoproteins. In glycoproteins, guys, remember, I'm just telling you the percentage difference. In glycoproteins, the glyco, that is the carbohydrate part. The carbohydrate part, it is basically 5%. And the protein part, it is basically 95%. It is basically 95%. Right? It is basically 95%. Now, over here, just try to remember how the proteoglycans are formed, right? Now let me just draw a very simple diagram and just try to explain you how a proteoglycan is formed. Now let us assume, guys, that this is, now let us assume like this is the protein. This is a protein, and in proteoglycans, basically the protein part, it is 5%, and glycosaminoglycans, it is 95%. Now remember, the protein is basically all the GAGs. Let me just draw the GAGs. Now let us suppose that this is a GAG, glycosaminoglycans. These GAGs are attached to protein. So remember, they are attached to protein with a stalk. Okay, with a stalk, right? So remember that they are attached to protein with a stalk. Okay, these are the glycosaminoglycans. They are attached to the protein with a stalk. And remember that, yes, this part, the part of the stalk, it is basically made up of three sugar, that is galactose and xylose. It is trisaccharide, right? And remember the shape of the proteoglycan monomer. That is again a very important MCQ question also, right? The shape of the proteoglycan monomer, it is bottle brush shape. Very important. It is bottle brush shape. It is bottle brush shape, right? So remember that the proteoglycans are the molecules that are heavily glycosylated. Proteoglycans, proteoglycans, remember they are, yes, heavily glycosylated, right? Meaning they are, okay, now when I say heavily glycosylated, it clearly means that they contain long, yes, linear means unbranched, linear chains of GAGs. And this is basically, they are attached to the core protein. They are attached to the core protein, right? Now, GAGs are basically, yeah, we have different GAGs, guys. You already know we have chondroitin sulfate, heparin sulfate, right? So, yes. Now, what is the biochemical significance of this, guys? Just try to remember that the proteoglycans helps in the structural support. Now, when I say structural support, they are major components of, they are major components of extracellular matrix. Okay. Now, they provide support to the tissue structure, right? They form a gel-like matrix that helps filling the spaces in between the cells, right? Maintaining the tissue integrity. They also help in cell signaling, guys. They also help in cell signaling. You already know the GAGs, especially I'm talking about the GAGs, that is 95%. Okay, GHEs that contribute to 95% of this proteoglycans. No, they can bind to a variety of signaling molecules, including they can bind to like very like molecules, signaling molecules, including the fibroblast growth factor. Okay, that is FGFs, right? And transforming growth factor, TGF, right? So this interaction basically regulates the cell proliferation, and you already know the cell will again after proliferation go undergo differentiation and migration, right? So yes, now especially they are functioning as the cartilage, right? The proteoglycan, especially if we talk about aggrecan, if we talk about aggrecan, it provides resilience and enables the tissue to withstand the compressive forces, right? The proteoglycans in the cartilage, right, play a very critical role in joint movement and reduce friction. They help in joint movement and reduce friction. Reduce friction. For examples, the proteoglycans you can remember, we have, yes, aggrecan. It is a large proteoglycan, guys, that is found in the cartilage, right? And it is essential for the tissue's ability to resist the compressive forces. Other more example that you can remember, guys, over here we have syndican. Syndican, it is basically it participates in cell adhesion, guys, or even in the cell signaling or cell migration also, right? So this was regarding the proteoglycans. Let us move on to the other part of the question, that is the glycoproteins. The glycoprotein. So these are the major ones that you can include these points in your answers and write. Just try to be more specific, guys, right? The other question that we are going to discuss it is, I mean, the other part in this question, it is the glycoproteins. The glycoproteins. Now, when I say glycoproteins, remember, guys, the glyco part, the carbohydrate part, it is only 5%. And the protein part, it is 95%. Unlike the proteoglycans. So these are the proteins. Now, remember that these are the proteins that is covalently. Covalently means by sharing. Basically, they're attached to carbohydrate chains. They are attached to carbohydrate chains, right? So they are attached to carbohydrate chains. Now, remember, there's a process, guys, called as glycosylation. You already know what does this mean. This means the addition of sugars, right? The addition of sugars to proteins. Okay, and that too they occur in the endoplasmic reticulum and the Golgi apparatus. Okay, glycosylation means the it's the process by which sugars are added to proteins. And the site where this glycosylation takes place, you can just remember it is the endoplasmic reticulum. Endoplasmic reticulum and Golgi apparatus. And Golgi apparatus. Golgi apparatus, right? Hmm. Now, what is the biochemical significance, guys? It also helps in what is the biochemical significance? Remember, guys, it also helps in cell to cell communication. Now, receptors on the cell surface. Now, imagine, guys, suppose this is a cell, and on the surface of the cell, we have these receptors, right? Receptors on the cell surface, remember, guys, often have glycosylated regions. They have some regions which are no rich in sugar, right? They have some regions which are called as glycosylated regions. And remember, these glycosylated regions, okay, help in recognizing glycosylated regions. They help in recognizing signaling molecules, signaling molecules, remember, such as hormones, cytokines, and growth factors. Hormones, cytokines, and growth factors, right? Now, for immune response, it also helps in, guys, the second one that you can remember, that is cell adhesion. Cell adhesion, this is again a question and I'm explaining you over here only. Now, many glycoproteins are involved in cell adhesion, where they participate in the formation of tight junctions. Tight junctions. Remember, they also can form adherence junction, right? And desmosomes also, right? So, for example, if we talk about, you must know about cadherins and integrins. Integrins, they are basically glycoproteins that mediate cell attachment to the extracellular matrix, right, and neighboring cells, right? So yes, the next one that you can remember, we have the protein stability and folding, right? Now, glycosylation can also help in no proper folding of the proteins. It helps in, or it supports in the proper folding of the proteins, and it actually no prevents the aggregation part. Okay, so yes, this ultimately no will no stabilize the proteins and other thing, right? One more important function that I want you to remember over here, that is, yes, the molecular recognition. The molecular recognition, guys, remember, the carbohydrate chains which are now attached to proteins act as recognition sites for various other molecules. For example, if you actually know about selectins, these are the glycoproteins which mediate the interaction between white blood cells and endothelial cells. So selectins are the proteins which are basically, yes, the are the glycoproteins. These selectins are nothing but these are the glycoproteins only, right? And they are mediating the interaction between the white blood cells and the endothelial cells, right, during the immune response. Example, glycophorin. Okay, example, we can have more glycophorin, right? RBCs. It is not about molecular recognition. The other one which you can remember, a glycophorin is basically an example of glycoprotein that helps in maintaining the RBC shape, right? We also can have erythropoietin, right? So it's a hormone regulating the red blood cell production. So these are the various things that you can remember about glycoproteins and proteoglycans for sure, right? So this was regarding the very first question. Now coming on to the second question. The second question says describe the role of glycolipids in cell membrane and cell signaling. I think we have already discussed this, right? So yes, let me just try to make it more easy. Glycolipids. Glycolipids. Now, when I say glycolipids, it's a class of lipids that have carbohydrate. Glyco means carbohydrate, that have carbohydrate chains, and they are covalently attached to these lipids molecule, right? So These glycolipids basically they are found on the outer leaflet of the cell membrane. Outer leaflet of the cell membrane. Okay, and they play crucial roles in structural integrity and support. Now, what is the role of glycolipids in cell membrane? So remember, guys, one is called as the membrane asymmetry. Membrane. So basically, remember, guys, that the glycolipids they are located outside or on the outer surface of yes, the outer surface of the cell membrane, right? Or extracellular surface of the plasma membrane. So yes, the hydrophilic carbohydrate chains extends into the extracellular space. For example, you already know if suppose this is the membrane, and yes, the glycolipids are basically attached to yes the outer surface of the membrane, and they're basically going into the extracellular space over here. The outside space of the cell it is called as the extracellular space. Okay. So this arrangement helps maintain the asymmetry of the cell, right? Which is important for cell interaction with the environment. Obviously, if the glycolipids are over here, they will be more interacting with the environment. Okay. They also help in membrane fluidity, right? So yes, now fluidity means where the movement is very very important. No, for okay, the fluidity is very very important for the movement of no proteins and other lipids. Okay, within the bilayer. So the carbohydrate groups they are also helping in stabilizing the membrane structure by preventing the membrane to become too rigid, okay, or too fluid, right? So that is helping in membrane fluidity and stability also. The third one, it helps in the formation of lipid rafts. Formation of lipid rafts, right? So they play a key role in formation of lipid rafts, right? Microdomains within the membrane that are enriched in cholesterol, right? The sphingolipids and glycolipids. Okay. So cell signaling, guys, you already know we have just discussed. Yes, the cell signaling. Right. So glycolipids are involved in the recognition of cell and non-self immune response. For example, glycolipids on the certain no surface of the immune cells. Okay. Glycolipids on certain surface of the immune cells. Now, they serve as a marker for recognizing other cells during processes like antigen presentation. Guys, you will be discussing this antigen wala part in pathology or in immunology later, right? The interaction between glycolipids and lectins. Lectins, it's basically the carbohydrate binding proteins. I will be discussing this yet. Can trigger the signaling cascades, molecules that regulate the immune cell activation, okay, and migration. So that was regarding this question. Coming on to the next one, that is the biological importance of glycosaminoglycans and involvement in cellular processes. So remember, guys, the major glycosaminoglycans that you have to remember. The major glycosaminoglycans that you can remember over here. We have chondroitin sulfate, chondroitin sulfate. We have keratin sulfate. We have heparin sulfate. We have dermatin sulfate. Dermatin sulfate. We have heparin. We also have hyaluronic acid. Hyaluronic acid. Now, what is the importance of the glycosaminoglycans and the involvement in cellular process? For example, let us just start with the chondroitin sulfate. Chondroitin sulfate, most abundant GAG. Okay, it is the most heterogeneous GAG, glycosaminoglycans. It is responsible for, remember again, a very, very important MCQ guys over here. It is responsible for compressibility of cartilage, yeah, sorry for the interruption. It is responsible for the compressibility of cartilages and weight bearing tendon. Now remember this chondroitin sulfate, it is basically seen in cartilage. It is seen in bone. It is seen in central nervous system, right? Second one that you have to remember, we have the keratin sulfate, right? Now keratin sulfate, it is basically formed from N-acetyl. I told you, the glycosaminoglycans will have the sugar, right? N-acetyl glucosamine plus galactose, right? So basically we have two types over here, two types, keratin sulfate 1 and keratin sulfate 2. Now keratin sulfate 1, it is basically isolated from cornea. Okay, and keratin sulfate 2, it is found in the cartilage. Okay, remember that it is present in loose connective tissue also. It is present in loose connective tissue also. Okay, and one of the important question or MCQ that they can ask you, remember that the keratin sulfate, it is responsible for corneal transparency. It is responsible for corneal transparency. One more MCQ that they can ask you, it is the GAG with no uronic acid. No uronic acid. I told you the acidic sugar is also there, but over here it is the GAG, the glycosaminoglycan with no uronic acid, and these two are very, very important questions that can be put up in MCQ as well. Right. Coming on to the third one, guys. So yeah, the third one that we have to discuss, that is the dermatin sulfate. Let us first focus on dermatin sulfate, though it is very easy to know discuss. The third one, that is the dermatin sulfate. Dermatin sulfate. Remember, it is widely located GAG. Okay, and remember, it is the most abundant GAG in skin. It is the most abundant GAG in the skin, right? So that is again a very, very important MCQ that you have to remember, right? So it's an important MCQ that has to be remembered from the exam point of view. The fourth one, remember the keratin sulfate. Not keratin, just yes, the heparin sulfate. We have already discussed the keratin one. Yes, the heparin sulfate. Now, remember, guys, the basement membrane of renal glomeruli. You already know the basic structural unit or the cell of the kidney. It is the nephron, and nephron, the part of the nephron, it has glomerulus, right? So the basement membrane of the renal glomeruli, basically what is present? Heparin sulfate is present. Now, you already know, according to your physiology knowledge, that the renal glomerulus, it is having negative charge. It is having the negative charge. Now, heparin sulfate, it is responsible, again, a very, very important MCQ, it is responsible for charge selectiveness. Charge selectiveness of renal glomeruli. Of renal glomeruli. It is responsible for the charge selectiveness of renal glomeruli. For example, guys, there is a question that has been asked multiple number of times. Why not albumin? It is coming into the renal filtrate. Now, guys, you already know that albumin, it is having negative charge. Okay. And heparin sulfate, which is present in the basement membrane, it is also having negative charge. Now, both these negative charge will repel each other. That's why albumin is never coming out in the renal filtrate, right? So that is something you have to remember. Now, one more that you can remember, it is the hyaluronic acid. Now, it is formed from N-acetyl glucosamine plus glucuronic acid. Remember that it helps in cell migration. It helps in cell migration. The very first one, that is called as tumor cell metastasis, morphogenesis, and wound repair. And wound repair, right? So yes, one more GAG that you can remember, we have written over here also, that is the heparin. Heparin, guys, remember again, they have asked one question. It is made up of glucosamine, plus ioduronic acid. Ioduronic acid. Remember, heparin, it is an anticoagulant, and it is the only, again, a very, very important MCQ, it is the only intracellular GAG. Intracellular GAG. It is basically present inside lungs, present inside mast cells, and also lungs. Important, right? So that was regarding the second question of the short answer type, right? The third one, guys, that is the concept of lectins and their function in cell to cell interactions, right? Now, let us start with this answer. Now, what are lectins? First, we have to understand what are lectins. Now, lectins are nothing but they are the proteins. Remember their composition, they are the proteins that have the ability to recognize and bind to specific carbohydrate structures. Specific carbohydrate structures, right? Now, specific carbohydrates, basically, if we are talking about glycoproteins, I'm talking about glycoproteins or glycolipids. or glycolipids. Okay, and these glycoproteins and glycolipids are on the surface of cell or the biological molecules, right? So yes, now this binding basically it occurs between the non the non covalent interactions, right? So yes, now when I say lectins, guys, they are not catalyzing any particular type of reaction. They do not catalyze any biochemical reaction. Okay, unlike enzymes, so they do not catalyze. Remember, do not catalyze any biochemical reactions, unlike enzymes, right? So we have various types of lectins, right? The very first one, the types which you can remember, although it's not a part, but you should remember, we have C-type lectins. So basically, they're classified based on their structure, guys, and the types of carbohydrates that they bind. So we have like, yes, the C-type lectins, right? The lectins basically require calcium ions for carbohydrate binding. Okay, for example, mannose binding lectin, MBL, mannose binding lectin. We have dendritic cell specific intercellular adhesion molecule 3 grabbing non-integrate. Again, a tough name, but if you can remember, it is DC sign, right? We have P-type lectins also. We have P-type lectins also. Okay, so basically the lectins recognize phosphate containing carbohydrates such as for example, you already know galactose 6 phosphate, right? So they are involved in carbohydrate metabolism, right? We have L-type lectins also. L-type lectins also. So basically, it helps in the recognition of galactose or N-acetyl galactosamine, right? Which are residues on glycoproteins and like glycolipids. So we have S-type lectins also. Again, S stands for the sulfated carbohydrates, right? So yes, they also help in cell adhesion and recognition. So these are the type based on the structure and the carbohydrate which they are binding. Now, the functions of lectins in cell to cell interaction, the very first one that you have to remember, it helps in cell adhesion. Okay, the cell adhesion, right? So basically, it helps in endothelial cell adhesion to the leukocytes, to WBCs, right? During immune response, any immune response, these endothelial cells will now come and bind to these, yeah, response. Will come and bind to these one, and especially if we talk about over here, we have the common example that is called as selectins. So selectins are subclass of C type. Okay, they are the major roles. For example, we have E selectin, we have P selectin, we have L selectin, right? They recognize specific carbohydrate structures, right? Celloics, right? X, they are basically located on endothelial on leukocytes, and know they facilitate their rolling. You'll be discussing this all in the like pathology part and the immunology, right? So that is something you have to remember, right? They also help in homotypic and heterotypic cell adhesion. Homo means same to same and hetero means different. They also help in cell migration also. Now they can regulate the migration by controlling. They can regulate the migration by controlling the, yes. interactions between the cells and extracellular matrix. For example, if we take about a case, now let us suppose there is wound healing, lectins can facilitate the migration of endothelial cells. Okay, endothelial cells, fibroblast, or any immune cell, and at the site of the injury, okay, promoting tissue repair and regeneration. So that is regarding the, yes, the lectins, the types, and the functions in cell-to-cell interaction, right? Now coming on to the next question, the very short answer type, 2 marks question. What are the blood group antigens, right? The blood group antigens, right? So you already know blood group antigens are distinct surface markers. Blood group antigens, they are distinct surface antigens, right? Or not surface antigens, actually, the surface markers. Okay, they are found on RBCs. Okay, on the surface of RBCs, right? So they are recognized by the immune system, and they are the key factors known in blood transfusion and everything. So you already know. Yes, so we have various now the carbohydrate structures in ABO antigens, right? We already know we have different blood groups. The ABO blood group system, right? We have, yes. For example, if we have type A blood group, right? Type A. You already know individuals with type A blood will have A antigen on the surface. It will have A antigen on surface of RBCs, now which are characterized by N-acetyl glucosamine, right? N-acetyl glucosamine, we have just discussed this, right? Right. Type B will have, right, B antigen, which are characterized by galactose. And galactose is nothing but the sugar only, right? How they are related to carbohydrates, right? So these are the sugar only. If we talk about type AB, so they both have A and B antigens on their surface, right? And we have type O individuals, they have neither A or B antigen, on the red blood cells. Okay, so that, remember, right? So yes, so these are the various carbohydrates which are now found in these various blood types. You can just remember that. Next question, guys, define glycocalyx and its role in extracellular matrix. We have already covered this earlier, right, in the first question itself. Explain how the carbohydrates are involved in cell adhesion. Again, I have already told you about the cell adhesion also, right? So we have discussed this, right? So yes, if we just want to get a summary out of it, guys, you already know we have lectin mediated adhesion. Now, when I say lectin mediated adhesion, means lectin means the carbohydrate binding proteins. Okay, the carbohydrate binding proteins. Okay, they bind to specific sugar residues on cells, right? They bind to sugar residues, right? For example, selectins, right? Now we have integrin. Okay, integrin mediated, we have already discussed this, integrin or cadherin. Okay, so these are the ones which help in cell addition. Integrin basically bound to carbohydrate containing extracellular matrix components. Okay, for example, collagen, or yes, we have fibronectin, right? So that is, you can just remember. So that was regarding the today's session, and thank you for joining and watching. Yes, thank you.